Today we're here with Dr. Victor Stora from the University of Pennsylvania, who's going to speak to us today a little bit about some genetic issues that have to affect our Lagotto Romagnolo. So Victor Stora, please introduce yourself and give us a little background.
Hi, I'm Dr. Victor Stora. I did my DVM at LSU for 4 years, and then I traveled to Virginia Tech for an internship in small animal medicine. And now I'm here as a resident in reproduction, medical genetics, and pediatrics. I'm sponsored by the AKC Canine Health Foundation, and the Theogenology Foundation. And I plan on getting a PhD in genetics concurrently after this residency.
Can you explain to us the terms of gene pool, genetic diversity, and genetic variety?
Sure. So a gene pool would be your total population of genes that you could use within a species, or more so in dogs, a breed. So it's basically all the potential that's there. Genetic diversity is how closely each one of those individuals is related to one another. So are they together more often line bred, or are they outcrosses where they are different lines from one another? And genetic variety would be the differences within a breed or differences within a population. So is there different color groups? Are there different coat types? Is there a difference in individuals that you would be looking for that would create more of an array of variety than just a standard dog? More like Rottweiler is very similar, whereas other breeds can have a very, very wide variety of coat colors and differences in coat, like Great Danes, for instance, can have a variety of coats that are allowed, which is a difference in variety.
Many of the diseases in Lagotto Romagnolo are inherited by an autosomal recessive mode. Can you explain the term autosomal recessive?
So, in autosomal recessive, we look at individuals that are bred together, and maybe you don't see disease in those individuals, and it ponders a lot of breeders because they breed these dogs and they think, oh yeah, that's a good pairing together because they're both very healthy and good representations of the breed. And then you get puppies that are sick, and it kind of throws you for a loop. And that's the hallmark of autosomal recessive. There's no sex predilection, so males and females are equally affected. The disease can come from completely healthy parents that were otherwise, normal. And the puppies may show disease at a typical age, so that it's a typical onset of disease is a hallmark of genetic disease as a whole. And I think that kind of throws people for a loop because they wonder why they're seeing disease in puppies that maybe, and the parents are completely healthy. So why am I doing something wrong? Is it me? Is it my choices? And it's not necessarily your choices. It's the stuff you can't see, the genes behind it that you can't see that these diseases will then come out. But luckily with recessive is you can choose proper pairings, and we can, as a whole, over a very long time, breed out a disease to where you do not see it anymore. But that's once a test is created, and once we kind of go through all of the forefront of testing and breeding and testing and breeding, we make that bad allele, which is a variant of a gene. So a gene is an area of the chromosome where you have a specific blueprint for something, say eye color or something along those lines. The alleles are the variants of that, so the different eye colors, the blue, the brown, the green. And there are differences even in necessary cellular function genes and anatomic genes that we have that can be either good or bad. And with recessive diseases, there's a good copy and a bad copy, or a proper functioning copy and a poor functioning copy, or a copy with a mutation in it. So that copy that is then passed on and on in generations, as long as it never sees itself again, will propagate no disease. Once it sees itself again, they don't have that backup copy, that good copy to make that proper functioning protein to live. And heterozygotes, or animals that possess a good copy and a bad copy, won't be diseased because they may need just 50% of that protein functioning, or the disease, what the gene transcribes, what the gene makes, to live. And they will live normal lives, and you will never know because you can't test for that protein to say that he has 50% less. So that's when we have the gene tests come in.
The breed is affected by several neurological diseases, and at this point, we have BFJE with a prevalence of 28%. We have Lagotto storage disease, which is at 8%, and then we have NAD, which is unknown at this time, but only carriers have been identified. Can you explain why it is important not to immediately remove all of the carriers from our breeding programs, and how do breeders move forward in lowering this percentage?
So the reason why carriers are so important is because if you look at the statistics for each one of those and they report the prevalence, they should also report the hets, the carriers, and the normals and the affecteds, and you'll notice that the majority of the population can be carriers. So if you remove all of those dogs, which what I previously said was they have half a function, they have a good copy, so they can live normal lives and you would never know that they're diseased. They then make up a majority of the population and are good stock to continue to breed. If you remove all of those, we go from a gene pool that is maybe more wide to an even more limited, narrow gene pool or a potential of genes, and the chances of unmasking further disease are there. Because when you limit the gene pool, then you have these animals that have inherent mutations in other genes that would otherwise go unnoticed. And you're breeding them more and more and harder in a line, and those bad copies then show up with other diseases, and you put yourself into a further hole because everyone's even further related to each other. So we want to breed cautiously and talk about how we can lower the risk of breeding, and that's when genetic testing comes in, because then we have hard proof of what that animal has underlying in its genes, and we can breed them with intellectual and breed them with a good thought process that our breeding is a good pairing and our risk is lower. When we start eliminating all those carriers, then new diseases come out. There won't necessarily be tests for those diseases right away, or even in the future if they're very complex diseases, so then it becomes even harder to breed with purpose and intellect. And I think you lose many lines to that because they breed hard on a line. They introduce more disease than they can take on, and then they have themselves pigeonholed where they can't outcross enough within the breed to make their line come back and be fertile again.
Okay, so what you're saying is if you take out too many carriers from breeding programs, is that you potentially can introduce, because you're limiting the gene pool, introduce more diseases into the breed.
Correct. No, there has been a paper that was done by Claire Wade, and in her paper there was a line that said no animal is inherent free of risk. And this is true. Everyone, even humans, have mutations in our genes that we have two copies. So we have one potential bad copy and one potential good copy, and we run around every day because we have that good copy to go back on. And we as humans outcross often, we're not very limited. Dogs we select based on specific criteria of what we were looking for, and with breeding we want predictability, similarity, and we want to be comfortable knowing that our litters are going to come out same predictable and with our kennel mark on it. So with that predictability of breeding comes, we introduce that bad allele again and again and again, and it has a chance of seeing itself again generations down if we use a popular sire for say. Who knows what is underlying in his genes? Who knows if he doesn't have something maybe related to cardiomyopathy in his background that you can't see, and maybe would largely be diluted out if he was allowed to be a dog in the wild. He would mate with everybody, and then it would not come back. His offspring wouldn't often come back to itself. But being breeders, we want that predictability, so we pull him back in over and over again, and that bad copy has a chance of seeing itself again and then creating disease. So as long as we kind of keep cognizant that the harder and harder we breed in a line, the more disease we may introduce, the more the individuals we eliminate or pull away from the gene pool, the more we're going to see as disease coming. The more you can keep in, and the more maybe minimal outcrossing you do within a breed, the more healthier your breeding program will be in the long run.
Okay, if a breeder were to have several, say, carriers in their programs of different genetic diseases, What advice would you give to them so that they could move forward with their breeding program and try to work towards eliminating the diseases in our breed?
So if they had a lot of carriers in their breed, I would almost mandate that their pairings have also the same high level of stringent testing that that breeder does. Because if he or she knows that they're carriers, they do their diligence in testing. So I would then use that as a springboard for other breeders to say, hey, I won't breed with that kennel or that unless you also provide the health testing that they're clear of disease, so that we know we're making healthy dogs in the long run. Because it's for the breed as a whole, and it's not for the individual breeder to then mandate that or say, hey, we need this, but it's for everyone's health to say, I would like genetic testing on your animals so that I can make sure that our pairing is healthy and that we're trying our best to eliminate as much disease as we can. And as you go on and on by picking these very, very stringent paired breedings, you then lower and lower that disease allele, because you'll be making hets or carriers. You'll also be making normals, and the normals will soon outrun the carriers in their ability to show promise for the breed, and eventually you won't be making carriers anymore. That allele falls off and it almost becomes non-existent.
Our genetic diversity right now sits at 35.4%. Would this be average to other breeds of dogs?
I would say that that's not uncommon. That I would not say that this is a particularly genetically limited breed in the grand scheme of things, because all breeds are limited. We take these, a population of dogs and we subsegregate it further, and then no one is allowed to breed with each other except for those that were in the original stud book. So again, everyone is now limited. So the practices that I talk about, or I can say quote unquote genetic hygiene, is using these breeds as a whole and determining proper schemes for everyone, and not just limited breeds, because there are even further limited breeds than a 35.4 that are more trouble than what you would maybe put yourself into. I don't think that that's too undiverse to related to one another.
Given that we have several neurological diseases and we have hip dysplasia, we have early onset cataracts, knowing that we have these particular instances of disease in our breed, what do you think is important to consider with breeding Lagotto?
So with all of, to be honest, that's a fairly healthy breed because of you don't have too many health disorders to worry about. So what I think I would put as the forefront of breeding Lagotto would be using exceptional conformation dogs and making sure that we have all of our health testing done, and the amount of disease that you have is somewhat limited. So I think that the full gamut actually can be run on most dogs and bitches, and you wouldn't get bogged down in all the nitty-gritty of some other breeds that have a plethora of health problems, that the further and further you look, the more and more you want to eliminate an individual, the more you limit the gene pool. I think that doing a minimal health testing that you have, like eyes, hips, and the coupled DNA testing is a perfectly acceptable route. Let's say a dog has somewhat, not dysplastic hips, but they're not the best hips, but they're a good representation of your breed, and they're a het or a carrier for BJFE, so we can know that we can breed that dog to a normal with better hips, and we'll improve our hip score slowly over time, and we'll slowly start reducing that allele frequency of the aberrant epileptic allele over the course of a long term. So just doing your diligence in testing, using forethought. I think breeders think too close in the future. My next litter. It's not your next litter. It's 4 or 5 generations down that you're going to see the problems if you don't do proper testing, or if you start eliminating carriers off the bat. It's going to be the future of the breed that doesn't benefit from it. And I think that you need to think futuristically as a breeder rather than thinking in the present, because I know a lot of stuff goes on in the present. You have a litter of puppies, you have this going on, and you just want to find your next pairing. But do your diligence, because that's going to affect you in the future, 4 or 5 generations down, not immediately.
Thank you very much, Dr. Victor Stora. for joining us this afternoon. You've given us some very helpful information, and I hope we can move forward in our breeding programs and learn something from what you shared with us today.
Thank you for having me. It was my pleasure of doing this. I really appreciate helping you guys out.